See things. All right. Hopefully we can see
Okay,
the proper
perfect.
side of this. Beautiful, beautiful, beautiful.
Yes. Okay. You
All right.
can you can start, Michael.
Are we
Welcome.
set to go? Okay, wonderful. Thank you, Jorge, and thank you so much for the invitation. Thank everyone for permitting me to talk about how I've built an undergraduate program in astrobiology here at Dakota State. It has been several years of tough labor, but we have made a lot of progress here, about which I'm super proud. So the front slide here is quite busy. Lots of students who have come through my first prebiotic chemistry class ever offered here at Dakota State, in which we engaged in original research topics, and we sort of designed the class to be very much in the spirit of a graduate laboratory situation for graduate students. And I would like to talk about this and talk about how I started here at Dakota State by first teaching students and teaching the first classes here, and then how we morphed all of this into what has become now a fairly vibrant and nicely dynamic research program here in astrobiology. I've got lots of folks to thank, including the Instituto de Astrobiología de Colombia, for sending me some brilliant undergraduate students last year. And so they have contributed immensely to the development of my program. Several other undergraduates, this is all undergraduate work I will be sharing with you today, thinking this might be a sort of a novel way to approach this from perhaps an education meets research standpoint. Want to thank my collaborators from Laurie Barge's group and Jessica Weber, who will be presenting here later as well, Bethany Dealing at NASA Goddard, and my longtime colleague Patrick Badeau, who could not join us today at Southern Oregon University. And certainly we have some great people to thank at NSF and South Dakota EPSCoR for paying a lot of the bills here as well. So we're very excited to move forward. So a little bit about how I got started here. When I was recruited to Dakota State, this is a school that really didn't have any science programs other than support programs for the other larger degree programs. Dakota State is known for its cyber defense, cyber operations, and increasingly its computer science programs. And so the science programs were really anemic here. They were really only support infrastructural programs. When I came here in 2013, I was, of course, brought here to make a real undergraduate program and ultimately to bring this idea of an astrobiology focus to Dakota State University, where none had previously existed. So we want to get in here, and so the thrust of my talk is going to be about how I kind of touched down here at Dakota State with really nothing, no resources other than basic teaching supplies, and started trying to immerse my students in real research problems in a class setting. So I initially set out to teach a general education astrobiology course in 2016. And that was very general, not really research-based, but just to kind of get the proverbial ball rolling, to get students interested, to let students know that Gaylor was here and that he was preparing to begin offering some of these sort of neat classes that had never been offered before. In 2018, I then stepped this up to a majors course in prebiotic chemistry, where all of the students had to have all of the majors prerequisites before they could get into the class. And so I used that class to really flesh out the beginning of an undergraduate research program here at Dakota State. And so again, the course was cross-listed in chemistry and biology. The course was entitled Prebiotic Chemistry. And again, it was taught very heavily in the spirit of a graduate class. I took a handful of students who expressed PhD level ambitions to me, enrolled them in the class, explained to them that they were going to be functioning as graduate students just like they would be required to do once they got out into the real world of graduate education. And that's how I started the class. So I only ended up having about 5 students in the class doing things, which made it very, very nice for hands-on research. And they presented weekly. We did laboratory chalk talks. We led discussions of the primary literature, students taking lead on this. But more importantly, I started immersing the kids all in real hands-on research that address novel problems. And that's kind of how we started here. Just for those who might be interested, and I know there are some in my session here who are also teaching it primarily in primarily undergraduate situations, most notably Jay Forsyth. So I think he probably can relate to some of what what's happening here. And so I put together the first syllabus here, just a couple of pages of it here just to illustrate sort of a typical course syllabus here. Cross-listed the class in our biology major as well, because I was hoping to get students that had from the biology major who had some interest in chemistry as well. I managed to get a couple of them to jump in. And as you can look here for the tentative class outline and schedules, everything is very loose. Read papers, discuss papers, go into the lab. And in this case, we were doing a lot of work with developing methods for assessing how polycyclic aromatic hydrocarbons are converted under various simulated prebiotic conditions to produce biologically interesting molecules. So I started the class this way and was really winging it. I had no guidance on this. I just jumped in and sort of started doing it. This led students to, once they got in and started reading the literature, most of the students got very, very excited about this concept of the aromatic world hypothesis, which I'm fairly confident everybody in the session knows something about. And the role of minerals in origins of life. And this is a thing that many of us have a great deal of interest in. I had always been into this pH world hypothesis idea, even going back to my graduate training. I finished my PhD in 2010, and I was really starting to get very much more interested in moving down this path. So I sort of pushed the students in this direction, and they got very, very excited about this. When they learned that pHs can be converted in meteoritic systems and asteroidal systems into quinones that could be biologically active, this became the focus of the class. The students wanted to go in this direction, so I set them about doing various analytical things, developing very simple analytical methodologies, experimental methodologies, emphasis on cheap, as I am very much an under-resourced program here, to address some of these real-world issues in hopes that we might tease out some data that had some publication quality that might contribute to the field. So students ultimately ended up in this class. We did some preliminary stuff. We found that we got nice conversion of a representative PAH molecule, in this case anthracene, conversion to a 9,10-anthraquinone, which we know has some very important biological features in contemporary life. So we got very excited about this. Others had shown that this molecule can be produced from PAHs, but no one had beforehand shown this in the context of origins of life. So we then focused on incubating anthracene under these simulated conditions to produce the major product, which was the 9,10-anthraquinone. And so off we went sort of to the races on this. We started generating some very preliminary results. Students made mistakes, of course, because they were quite inexperienced. But by the time we got a few weeks into this, we were actually starting to get consistent results. We were incubating anthracene on various mineral phases. Primarily the montmorillonite was the one that was easiest to work with, seemed to be quite relevant enough, and it served as a great sort of training matrix for my students. We developed the GCMS method. We verified production of the anthraquinone from the anthracene in the presence of the montmorillonite. Because I lack an NMR device here in my lab, we had to verify compound production using co-injections and co-spikings and things like that, and comparable overlapping retention times, which turned out to be a great teaching method for getting the students to understand that when you don't have a tremendous amount of equipment, you have to go about this type of science in different ways. So in my lab, we're fond of sort of calling this the ghetto approach to doing things, but it was sufficient to demonstrate that we were in fact producing the compound that we know that we were saying we were produced. And ultimately, this generated a publication quality data set. We ultimately then were interested in kinetics. As it turned out, I had been teaching kinetics right before I convened this class in my general chemistry class. Those students in organic chemistry, or rather, I'm sorry, in prebiotic chemistry, they had to now recall all of that stuff that they probably tried desperately to forget during this time. They had to sort of remember what are rate curves and what are rate constants and what are the orders of reactions. And so we set about to map out the kinetics of the conversion of the anthracene to the biological anthraquinone, at a variety of temperatures that are relevant to a variety of astrochemical environments. We ended up generating, after hundreds and hundreds of samples and some student frustration, and why isn't this working? You know, this is a thing that students, you know, they get frustrated when they don't get the right result right away. This is a thing that we struggle with teaching undergraduates and training them. But after hundreds and hundreds of samples and getting our methodologies down, we ultimately were able to generate the curve that you see here, where we demonstrate production. We measure production rates. In this case, we confined our rates to the linear range to produce ultimately the curve that you see here, which is starting to head in a very much a sigmoidal direction here. And so we got a very, very interesting result here. We have this nice sigmoidal curve over, in this case, the best we could do with our equipment was up to 250 degrees C. So the students were very excited about this. I was very excited about this. We also wanted to try to get a little handle on mechanism. Again, we were a very under-resourced lab, so we had to get at this by confining the reaction mixtures in an air. Right. We initially did this in ceramic crucibles, which again is kind of a cheap way to do this, but it worked. Reaction efficiencies were very low, but we still got good data that increased with temperature and time and all those things you like to see with kinetic studies. We then looked at air in a truly sealed vial and showed production. No big sort of revelation there with molecular oxygen, which we think is the primary mechanism for production of the quinones, probably all of the quinones. We then measured CO2 and argon in sealed containers, and we saw no production. So we at least had a hint at what the mechanism might be under this experimental situation. So what did the class produce? The class actually worked very hard. When you have this was a 4-credit class, and so we had 3 hours of lab time a week and 3 hours of lecture time a week. Well, those hours began to blur together as we got deeper into the research. We were doing less in lecture and much more in lab, and I sort of knew this would be the way this would go, and I was fine with that. The students were engaged, they were learning a lot, they were really getting a lot of hands-on training. And so the ultimate deliverables for the course were, when I set the course up, would be at least a manuscript prepared according to the author's guide for, in this case, we selected Origins of Life, Evolution of Biospheres, some kind of presentation at a meeting, either our own Dakota State Research Symposium or the regional ACS meeting, and it turns out we got the kids to both of those, and then ultimately we got this paper put together with Laurie Barge and Bethany Dealing at NASA and helping us out, sort of fine-tune this stuff. And we got a nice paper in OLab. And so the kids were very excited about this being their first paper. And now, interestingly enough, and I'm excited, all these kids have now gone on to doctoral programs and med programs and things like that. So big success with this program. So from that, I wanted to take all of that success and I wanted to morph it all into what would ultimately be my more sustained research foci here at Dakota State. I'm classically trained as an environmental analytical chemist, so I did not get into astrobiological research until the early years of my first professorial job, so that would have been around 2012, 2013. And then these two classes were kind of, can I do this? Do I know what I'm doing? Am I capable enough to teach myself how to be an astrobiologist? Well, I feel like we were getting there. So after the paper came out, I was out hanging out on social media, and Jorge Bueno, the director here of the Institute, the Instituto of Astrobiología de Colombia, reached out to me and had this grand idea that we should set up some sort of exchange, and he was very excited about what we were doing, and we'd love to send some of our students up there for an immersive summer experience. And I was, of course, giddy about this. Excited that someone was sort of paying attention to the work that I had achieved with inexperienced undergraduates and the opportunity to bring students from another country and from another very cool institution to my lab for the summer to engage and step this work to the next level. Turned out to be very exciting. And I got two amazing students in Sarah Gonzalez and Vitas Karanauskas, who joined two others of my students who I had already recruited from a different funding source for the summer. And they worked together as an amazing team. But with the IAC students began really the birth of sort of a real astrobiology program here. So fast forwarding here a little bit before I start kind of getting in, what I want to do is survey basically results that the kids have sort of produced over the last 2 years. But jumping ahead almost a year after they worked with me for the summer and then went back to Colombia, I was fortunate to land a sub-award from a massive South Dakota EPSCoR, NSF EPSCoR grant, a $20 million grant that was awarded to the state of South Dakota. From that, I got $150,000 to support 25 students over 5 years engaged in immersive research in astrobiology. So that was a really, really big deal. And a lot of that, the recognition and the accolades from the work that I had done with the Astrobiology Institute of Colombia students really laid the foundations and got me involved in this much larger grant. And so now I have funds to bring gifted students and ambitious students to my lab. And so I'm jumping forward here about 9 months or so to kind of set the stage for where we've gone. And so this past summer, I ran an intensive summer REU with 4 students in this case, funded by the new NSF EPSCoR money. And so what I want to do is kind of roll all this into a ball and then take you all through some of the neat research outcomes that have been produced by these kids. Right. So ultimately, what is the question being asked? Building on all of the preliminary results, and it turned out to be robust enough results to get us a paper into OLAB with minimal peer review concerns. It was very, very, very warmly accepted. So that was very, very exciting. But when things work, you want to keep moving that ball forward. So the basic idea here is that we know polycyclic aromatic hydrocarbons, or PAHs, are everywhere in the solar system and likely the galaxy and very likely the universe. We know that they move and they impact and they are deposited into planetary environments where they undergo a variety of mineral catalyzed and UV and thermal catalyzed conversions into oxidized products. Especially ketones are the things of biological interest that we are most interested in. All of us know that ketones play a fundamentally crucial role in cellular metabolic processes in extant life. So we took all of that interesting work, we showed that the anthraquinone is produced, that the minerals catalyze it. We wanted to look at new mineral substrates, we wanted to look at some relevant to Mars, and ultimately show that these are viable pathways from effectively geo or astrochemistry into biochemistry. And this has become kind of the core focus now of my program here over the last 2 years. And again, my recent EPSCoR research fellows and the brilliant kids that came to me from the Astrobiology Institute of Colombia have moved my program forward in a very, very sort of huge way here. So we've been looking at, in addition to montmorillonite, we wanted to start looking at another phyllosilicate that has more of a one-to-one octahedral, tetrahedral relationship. In preliminary studies, we saw a very different production in kinetics on this, so we added that to the inventory. We also wanted to look at one of the Mars regolith simulants, soil simulants. And then later, I finally started getting hold of some other cool simulants, including an asteroid, a carbonaceous asteroid simulant, which we just recently finished the work on. So I'll be talking about some of those results and what we did with this. So the first thing we had to do is we had to take the first summer team that had the Astrobiology Institute kids in that team. And one of the first things we did the first day they got here, after we got them settled down and said, hey, welcome to this weird place called South Dakota that you may never want to come again. I don't know. They seem to have fun, but I'm sure it was quite culture shock for them. We had a method using these larger volume ceramic crucibles, which are not particularly efficiently closed systems, but it's all we had to work with. So we used them for the entire first study. So the first thing we tackled was method development. We need a more efficient system that gives us higher rates of conversion, greater yields, greater efficiencies. And so the team started breaking out. The team broke out into let me try to get rid of, so this is in my way here. So the team started figured out how to design their own glass tubes. We took basic borosilicate pipettes. We burned them. We bought propane torches and we burned these things into something that would actually work. And we carried out the previous study we published in OLEV using the glass tubes with the montmorillonite over the same temperature. And we saw some staggeringly better results. We see astonishingly higher efficiencies in the tube incubation method, which allowed us to get a handle on even smaller trace compounds that we were not seeing in the crucibles, because it turns out we were losing far too much of the reactants. So we started out kind of slow, but then we have this cool method that seems to work very well. Slightly smaller amounts of mass is being used as well. So this was the first thing we did. Now, using this new method, I unleashed the Astrobiology Institute team and their colleagues on 3 substrates. We looked at the montmorillonite, we looked at kaolinite, and then we looked at the quite different Mojave Mars simulate, indicated as MMS. And we worked to generate curves. And so these 3 curves here represent probably some 2,000 or 2,500 actual samples run. The kids were probably getting pretty burned out on doing this kind of work, but we know that lab work is tedious, but it pays big dividends. We generated these nice publication quality curves. We see the montmorillonite in panel A hitting a sigmoidal patch here. We see the kaolinite kind of getting there slower, but it's starting to get into that sigmoidal phase. And we've done all the curve fitting, and we have the calculations and everything for this, but it cluttered up the plot, so I didn't bother putting them on the plots. And then in panel C, the Martian soil remains quadratic, which we thought was very interesting. Why would it be that even at the higher temperatures, in this case now, I got some money to buy a better oven so we could go to higher temperatures. So we wanted to go to something higher that was a little bit more representative of maybe a hot meteoritic or a hot asteroidal type situation, or even a very hot planetary situation where meteorites were being deposited on the surface. And these are the fits that we've got. They tell a very, very interesting story. And so we started to hypothesize what the heck is going on here, right? So the sigmoidal curves in panel A and B with the thiosilicates suggest saturation of catalytic sites. Probably not a big surprise at these very high temperatures, right? But we also started thinking there's probably a considerable amount of loss of both reactant and product as this chemistry happens. So we got very interested in this. This became kind of a working hypothesis for us. We ended up calculating conversion efficiencies with the glass tube approach that was in the 2.9, 1.7, 0.9% range, which was 2 orders of magnitude increase over conversion efficiencies that we saw using the crucible. So we developed a method now that is vastly better, giving us better results and easier and safer to work with in the lab. So yay, go method development. This is always a thing we have to do. So we got very curious about this, and we wanted to address this situation of where are we losing things, how tightly are the reactant and is the reactant and the product or products, are they binding to these 3 substrates? So we wanted to do a production and loss curve. We decided to do this midway between our temperature regime at 250 degrees C. And here are the results that the kids got here in this plot. We see the montmorillonite really cranking up fast, very high catalytic potential here. Each of these points represents a rate calculated at the specific points in over the linear range, rather, is what I was trying to say. So you see the montmorillonite really starts kicking off, and by about 10 minutes, we're on the loss curve at 250 degrees C. The kaolinite, a little bit slower to get going, softer slope, and then about 20 minutes, we start losing that as well. So the kaolinite appears to hold on for about 10 more minutes under this fairly harsh condition. The Mars soil, on the other hand, ratchets up pretty consistently with the montmorillonite, but quickly starts to die off at about 5 minutes. And then we see this nice kind of quasi-exponential approach back down to the x-axis here. And we let this go for 60 minutes. At this point, we were starting to get down to close to detection. We had our answer. So it looks like that we are losing product at these higher temperatures, which again, not a huge revelation, but when you're working with inexperienced undergraduates and also nobody has really done this stuff, we were pretty excited about this result. we wanted to take this a little bit further. Let me back up here just really quickly. The other thing I was trying to say that is my slides are obscured here a little bit by the screen sharing menu that I should probably move here, is that we thought maybe we were getting production of secondary products, and we ran these samples with all kinds of exhaustive GC conditions, including a very expensive PAH column that in principle would separate, even out trace amounts. And we really never found anything but the core quinones. We'll talk a little bit more about some of the peripheral products we found as I get deeper into the talk here. But this is a cool plot. These data are all now in final review. This paper is heading out to Astrobiology here very, very shortly. So this stuff is kind of hot off the press. So I got very interested, we got very interested in trying to understand what is happening with the loss of the compounds. And can it explain the production curves that we were seeing? So we set about doing mineral doped studies over the temperature range that you see here, less finer detail, because we figured for loss kinetics it was okay to not as much labor doing 50-degree intervals as we've done for production curves. And here's what we see. We see the number one here, we're using the numerical designation because originally we were going to submit this material. We did submit this material to an ACS Chem journal, and they wanted everything in numerical representations for compounds, and we just left the plots in those forms. So 1 is the parent anthracene, 2, of course, is the product anthraquinone. So on the left panel there, we have we have loss of the anthracene, and on the Mars soil, we see it really starts to go away pretty fast. Everything's pretty consistent till we get up to about 200 degrees. Then the materials really start to show their different properties. By the time we get to 400 degrees, we're really losing like crazy off of the Mars soil simulant. And then the montmorillonite and kaolinite are certainly diverging, but they're losing things at a more gentle rate. So this seems to help us understand a little bit about why we get those sigmoidal curves. We also wanted to look at loss of the product, anthraquinone. So in panel C here on the right, we did the same thing, over 100 to 500 degrees temperature. You can see in this case, the montmorillonite loses the anthraquinone at an extraordinary rate relative to the other two. In fact, so extraordinary, we had to produce this weird sort of broken curve here to get all of them properly on the curve. So this break here means that obviously we've got much, much higher, faster losses of the anthraquinone product, the main product we've been focusing on at the higher temperatures on the montmorillonite, whereas the kaolinite in the Mars soil, we're all kind of chugging along at about the same rate of loss. So at first we thought, okay, great, we've shown probably sublimation. How excited do we get about this? And then we had a meeting one day and we started talking about, you know, this could be a really big deal. How the heck else are these materials going to get out into an emergent biosphere situation and be distributed around, say, a prebiotic planet, say a primordial Earth or an ancient Mars, if they're not being heated up either upon impact with the planet or as a result of sort of moving through the atmosphere at high velocities. So we are suggesting that this is a very interesting way that these compounds can be distributed on sort of a global basis to drive this chemistry in multiple sectors on a prebiotic planet. So that's kind of serendipity that came out of that. Let's see, how am I doing on time here? Okay. We got started about 15 minutes late, it looks like, so we'll get through this. So these data here are literally fresh off the press. I had these in Excel plots, and just last night I got these done up in Adobe, so they were a tiny bit sexier to look at. So these are a part of a different paper. This is the carbonaceous asteroid simulant that we have collaborated with the University of Central Florida's Center for Lunar and Asteroid lab there. They've been very cool about giving us these things, and so we carried out the same study this summer, doing more or less the same things, looking at production of anthraquinone over the temperature range as done before. Again, we get into this kind of sigmoidal production on the asteroidal material, but if you look at the y-axis, we're seeing significantly lower production rates, lower quantities being generated relative to the other three materials. So we found that to be quite interesting as well. As done before, we were curious about loss rates and how that might explain the sigmoidal curve. So we looked at loss over 500 degrees in 100-degree intervals here, and we see that the anthraquinone is lost at a considerably higher rates and amounts relative to the parent anthracene. So this is a whole different story now with this carbonaceous so-called CI simulant. We're just calling it AST here for simplicity. So this thing is behaving a little bit differently than the others. So this is shaping up to be an interesting piece of this puzzle that we've generated here. The other thing that the kids found as, once we started, so we tailored our chemistry so that we were looking for the anthraquinone and all its molecular ions for every run to get proper quantitation and statistical reproducibility and all that sort of stuff. But after a while, I started getting curious about what other things were likely living in our solvent extracts. We were running solvent extracts at about 1.5 mils to 2 mils for a variety of reasons I'm not going to bore you to tears with. It just kind of worked that way. So we started concentrating these things down into 100, 200 microliter solvent volumes and running them, and we started picking up all of these other lesser produced products that I've made a little sort of what I'll call a ghetto table here. And we find these other things. These other quinones have been showing up in our samples. They are tentatively ID'd. We would, of course, need NMR. I've been talking to Lori Barge and Jessica Weber about getting some NMR work done with those folks out at NASA JPL. So we talked about that very recently, in fact. But we were excited to see that 4 of the compounds that we find were also recently reported by David Deemer's group at the University of California, where they showed that these quinones detected in meteorite extracts actually facilitated proton gradients across lipid membranes. And so we got really, really excited about their result. And of course, young undergraduates got super, super, super excited about seeing that their OLED paper had been cited by David's team there. And so everybody was like, yay, we've arrived. Dr. Deemer's group has said something about our work. So this was very, very, very cool. Anyway, tentatively ID'd stuff. We also see lots of the dimers being produced. That's a whole different study that I do not have time to get into today. But we've also been mapping out the kinetics of these dimers, the 9,9 bianthracene and biphenanthrene, because they fit nicely into the pH world hypothesis model. They've been shown to stack. They could, in fact, be able to behave like sort of prebiotic protonucleic acids. So we are quite interested in those as well. Some very one-off studies that I had the kids kind of break away and do towards the end of this past summer. And actually, I had Sarah Gonzalez and Vitas, my students from the IAC, last summer kind of start this work. And then I had my students this summer repeat it just to see if we got similar results. We wanted to start looking at some other planetary relevant minerals. And so we took our mid-level conditions, 250 degrees C, and we ran aluminum oxide, magnetite, hematite, iron phosphide, iron sulfide, and of course our clays, our MMS, and our asteroid. And what we see here is these oxides, perhaps not unexpectedly, produce pretty substantial amounts of anthraquinone under these conditions. The iron sulfide and iron phosphide were really interesting because we got almost nothing. We saw nothing with iron phosphide. We see almost nothing with iron sulfide under the same condition. So we really feel like we've got some mechanistic thing going on there that has to be vetted out. Again, these are super preliminary. I had the students do this as a one-off to see how well these materials behave and could we actually get reliable data. So this is definitely a work in progress at this point. We also, my conversations with Lori Barge at JPL, she has been awesome at grounding me in the world of planetary relevance. So conversations with her, many of those over fine wines before COVID took over, and then Zoom lines after COVID, suggested it was time for us to start exploring some of the more relevant Mars relevant minerals. So we got a bunch of these things. We looked at some of the perchlorates, nitrates, sulfates, phosphates, these things that have been reported being found in a number of different configurations and concentrations around the Martian surface, and we started to see if we could work with these. Many of them turned out to be really hygroscopic and very difficult to work with under the conditions we have here in my lab, but we did get some interesting results in this kind of first series of one-off studies. We found that the aluminum nitrate cranks out the anthraquinone and also cranks out similar levels of these nitroanthracenes, which, they could be very interesting precursors for perhaps protonucleic acids or other types of protobiomolecules. So this is another avenue that we kind of pried open as a one-off study here with the kids that I've been working with from these 2 summer REU programs. All right, these are things that are up on deck. My collaboration with the folks down at the University of Central Florida in the so-called class lab, they've been very generous about sending me several of these Mars relevant simulants now that represent higher priority astrobiological exploration missions now on the surface. I have yet to have the student power to really get on these 4, but we do have them in the lab. We've done some basic work with them. I'm now waiting for sort of my next batch of students to be psyched about kind of jumping in and repeating some of the studies we've done on these materials. So they're kind of on deck and waiting for my next crop of students, whoever that happens to be at this point. In the interest of saving us a little bit of time and getting our session here back on track a little bit, my student Sammy Drummond is he's one of my NSF EPSCoR research fellows. He worked with us over the summer. And yes. Oh, okay. Yeah, I can finish up. Not a problem. Thank you. I tried to make it so I left as much time as possible for questions, but we did get started late. So anyway, Sammy, Sammy Drummond came in. He's one of our international students from Australia, and he'll talk more about this in his own talk later in our session. But he came to me one day and said, "Hey, I'm kind of interested in what UV does. Can I do like a one-off study?" So he put some stuff outside, and we looked to see what production happened in the sun. He's going to talk about all that, but at the risk of stealing too much of his thunder, this has led to an entirely new avenue of inquiry in our lab, and Sammy's been driving all of this into the new academic year. And so I wanted to just kind of quickly share with you some data that he'll be presenting some different data, but we have been examining anthraquinone production on these different mineral substrates under various UV regimes and under various Mars atmospheric compositional regimes. And so there's a lot of data here. I don't expect to walk you through all of it, but I simply wanted to show that this is the next place that we have gone. This was supposed to be just a one-off thing to see what happened. Sammy's initial results were beautiful, and that has led us down this, what I like to call rabbit hole, into doing a whole series now of UV mineral catalysis studies. And we're getting some really, really nice results here, showing differential production in more Mars-relevant atmospheric and mineralogical circumstances here. So Sammy's going to talk a little bit about that in his talk later. In the interest of time, and I am not one for brevity, so I am clapping high five to myself for getting through this with leaving a little bit of time. Lots of people to thank. So excited to be invited to participate in this event. I, like everybody else who has presented, was very sad that I could not go down and hang out in the beautiful city of Barranquilla. So I'm very sad about that, but I'm very, very excited that the Institute and the Universidad de Atlántico were able to pull this thing together and make it a meaningful experience. And of course, I want to thank all of our current collaborators and certainly our funding agencies. And with that, I will take questions. Thank you. Yes. Okay. Okay, sure. Yes, I know. And I wanted to say something about that, Yvonne. That's a very good question. When I met Jorge, let's see, this would have been, been a couple of years ago now. And then we met at AppSciCon 29, and we talked a lot about trying to get more of a collaborative thing going. He continued to emphasize to me that undergraduate research had historically not been something that was really done in the Colombian system and perhaps other South American nations. And that was really news to me. And I felt that's kind of unfortunate because though you have to work harder with undergraduates often, the payoff for working with undergraduates when you finally get them to a point where, they are able to help you author a manuscript and go with you to a major conference and present. It's been a very, very satisfying thing for me. I always wanted to do this as a PhD student. I was the only student in my entire wing of my graduate program who wanted to work with primarily undergraduates and go the primarily undergraduate institution. I'm very research minded. Even as a graduate student, I was always mentoring the undergraduates that came through our laboratory for the summers, and I just developed passion for that. And the reality is that at my university, I have no graduate students, I have no postdocs, so I have to work with undergraduates, and I have to make them be as awesome as they can and get them functioning at the graduate level.
Okay. The second question is, what are the inorganic compounds that you consider... Okay, sorry, some technical difficulty here. What are the inorganic compounds that you consider essential to be used with raw materials in biological processes in a lab?
For that question, are you asking why we have selected the mineral phases that we have worked with? Is that correct?
Yeah, sure.
Yes. Yes. So originally, we, as I pointed out early in the talk, we got started. We needed to start simple, especially because I introduced all of this stuff into a class. And I knew that phyllosilicate minerals had catalytic capacity. Being trained as an environmental chemist and toxicologist, I knew that there had been a lot of use of these phyllosilicates to detoxify certain types of contaminants. And I thought this is a very interesting way for me to sort of springboard into my sort of newer interest in astrobiology. But we started with the simple things. We started with the clays. They were relatively easy to work with. They produced results fairly easily, not as painfully as we've encountered with some of the other mineral phases. And so we got the proverbial ball rolling. We got the program rolling on those kind of easier to characterize materials. That said, everything we've done so far, no one else has done. So this turns out to be novel. These have turned out to be novel exercises. But again, after meetings at AppSciCon and Gordon Research Conference and various conversations with Jorge and others at the Astrobiology Institute, we were starting to get the message that we needed to move this in the direction of those minerals that are more relevant for especially Mars exploration missions. And so that's why we've started to move in the direction that we have, especially with all the Mars simulants that are tailored for some of the very high priority astrobiology investigations. And so that's how we've gotten to the mineralogy that we have arrived at. I hope that answers the question effectively.
Okay, Michael, thank you very much. We are really grateful for having you in the 5th International Congress of Virtual Astrobiology.
Yes, thank you. Thank you.
We welcome you to come to Barranquilla and visit us, University of Atlántico.
I am very eager to do this. As soon as we make COVID go away, this must go away.
Okay, looking forward for it. Thank you. Have a great day.
All right.